Hello there, and welcome to Deep Sleep Seas. I'm so glad you found your way here tonight. Perhaps the day has been long, or maybe you're simply the kind of person who finds peace in the quiet mysteries of the ocean rather than counting imaginary sheep. Either way, this journey is for you. You have nothing to do but rest. Let your breathing slow, your muscles soften, and your mind drift gently downward, like a submarine descending through layers of blue darkness, deeper and deeper, until you reach a place few have ever seen. Because tonight we're traveling to one of the strangest places on Earth, not on land, not in space, but hidden far beneath the waves. We're going to explore the still lakes that rest at the bottom of the ocean, lakes within the sea. Pools of brine, so dense and so toxic that they refuse to mix with the water around them, forming shores and surfaces in a place where there should be none. These are the brine pools, and beneath their beauty lies a world of contradictions, a realm where death and life exist side by side, where ancient geology meets modern microbiology, and where the very rules of the ocean seem to bend. You'll hear of pools that preserve the dead for decades, of bacteria that thrive in conditions no animal could survive, of mussels clinging to toxic edges, and of scientists peering into windows to Earth's distant past. Some of what you hear tonight may surprise you, but mostly I hope it will carry you gently into sleep, drifting through these hidden depths like a slow current. Now let's begin. Far below the surface of the ocean, in places where sunlight has never reached, and where the weight of water presses down with crushing force, there exist bodies of water that seem to defy all logic. They are lakes, not lakes on land, but lakes on the seafloor. Pools of water, sitting within water, as strange as that sounds. If you could descend in a submarine, thousands of feet down through the cold and the dark, you might suddenly come upon a sight that would make you question your eyes. There, in the beam of your lights would be a shoreline, a boundary, a surface. The water below would shimmer differently, denser and thicker, with a visible edge where it meets the ocean above. You could see ripples moving across this underwater surface, disturbed by deep currents the way wind disturbs a pond. And if you tried to descend into it, your submarine would not sink smoothly, but would feel resistance, as though landing on something semi-solid. These are brine pools, bodies of hypersaline water. Water so saturated with salt and minerals that it is far denser than the seawater surrounding it. And because of this density, it does not mix, it pools. It collects in depressions on the seafloor, and stays there sometimes for thousands of years, forming what scientists call underwater lakes or deep hypersaline anoxic basins. The salt content in these pools can be 3 to 8 times greater than normal seawater, where the ocean around them contains about 35 grams of salt per liter. The brine pools can contain 200 grams or more, sometimes reaching concentrations that rival the Dead Sea. This extreme salinity creates a density so great that the boundary between brine and seawater becomes a physical barrier, almost like a liquid membrane. Scientists call this boundary the halocline, a transition zone where salinity changes dramatically over just a few meters. And it is not just salt that makes these pools unique. Many of them are also anoxic, meaning they contain almost no oxygen, without the mixing currents that carry oxygen down from the surface. And with microbial activity consuming what little oxygen seeps in, the brine becomes a suffocating environment. It is also often rich in chemicals that would be toxic to most life. Methane bubbles up from deep beneath the seafloor, along with hydrogen sulfide, the gas that smells like rotten eggs and is lethal in high concentrations. Some pools also contain elevated levels of heavy metals like iron, manganese, zinc, and copper, dissolved from the rocks and sediments below. The temperatures vary wildly depending on how the pool was formed. Some brine pools are cold, just a few degrees above freezing, sitting quietly in the deep like frigid ponds. Others are geothermally heated, their waters warmed by volcanic activity deep beneath the crust. The hottest brine pool ever discovered, the Atlantis II deep in the Red Sea, reaches temperatures of 68 degrees Celsius, hot enough to scald your hand. It sits in complete darkness, thousands of meters below the surface, radiating heat like a hidden furnace on the ocean floor. These pools range dramatically in size. Some are tiny, barely a meter across, little pockets of brine tucked into cracks and fissures. Others are vast. The Orca Basin in the Gulf of Mexico, the largest known brine pool on Earth, covers an area of 123 square kilometers and contains brine up to 220 meters deep. That is deeper than most skyscrapers are tall. It is a lake the size of a small city, hidden more than 2,000 meters beneath the sea. The story of how these pools form begins not in the present, but deep in geological time, millions of years ago. To understand brine pools, you must first understand salt. Not the salt you sprinkle on your dinner, but vast deposits of it, layers kilometers thick, buried beneath the ocean floor. These deposits are the remnants of ancient seas that once covered parts of the Earth and then vanished. During certain periods in Earth's history, shallow seas became cut off from the larger ocean. Tectonic forces, the slow grinding movement of the planet's crust, would close off basins, isolating them. Without a connection to the open ocean, these trapped seas would begin to evaporate under the sun. As the water disappeared, the salts dissolved in it would be left behind, precipitating out and settling on the seafloor in thick layers. This happened over and over, sometimes for millions of years, until layers of salt, gypsum, and other evaporite minerals had accumulated to staggering depths. One of the most important of these periods occurred during the Jurassic, around 150 to 200 million years ago. At that time, the Gulf of Mexico was a shallow, restricted sea. It dried and refilled multiple times, leaving behind a layer of salt called the Luen salt, which in some places is up to 8 kilometers thick. 8 kilometers, that is 5 miles of solid salt buried beneath the sediments that later covered it. In the Mediterranean, a similar but even more dramatic event occurred much more recently. About 6 million years ago, during what is called the Messinian salinity crisis, the connection between the Mediterranean Sea and the Atlantic Ocean was severed. The Mediterranean, trapped and evaporating under the hot sun, shrank dramatically. Some scientists believe it nearly dried up completely, leaving behind a landscape of salt flats and desert basins where a sea once was. Over hundreds of thousands of years, salt accumulated in layers across the basin floor. When the connection to the Atlantic was restored and the sea refilled, these salt deposits were buried under new sediments. In the Red Sea, tectonic activity and volcanic processes created similar conditions during the Miocene epoch, around 20 million years ago. The Red Sea sits on a divergent plate boundary, where the African and Arabian tectonic plates are slowly pulling apart. This rifting created a narrow, isolated basin that periodically evaporated, depositing salts that now lie deep beneath the seafloor. These ancient salt deposits are the key to modern brine pools. They sit there, buried and compressed, until something disturbs them, and that something is water. Seawater is constantly moving through the crust, seeping down through cracks and fissures, penetrating deep into the layers of sediment and rock. When this water encounters the buried salt, it begins to dissolve it, just as water dissolves table salt in a glass. The result is brine, a solution saturated with dissolved salts and minerals. This brine, now much denser than the seawater that created it, begins to move. In some places, it is pushed upward by pressure from below, squeezed out through faults and fractures like toothpaste from a tube. In other places, the movement of the salt itself creates pathways. Salt is not rigid like most rock. Under pressure, it flows slowly and ponderously, like a very thick liquid. This process is called salt tectonics, and it is responsible for creating many of the structures we see on the ocean floor. Over millions of years, the buried salt layers can be pushed upward, forming domes or diapirs that rise through the overlying sediments. Sometimes these salt domes reach the seafloor and are exposed directly to seawater. When that happens, the salt begins to dissolve and brine is formed right at the surface. The dense brine, unable to mix with the lighter seawater, flows downhill along the seafloor, following the contours of the bottom until it finds a depression where it can pool. This is how many of the brine pools in the Gulf of Mexico were formed. The ancient Luen salt, buried for millions of years, has been deformed and pushed upward by tectonic forces. In places, it breaches the seafloor and seawater dissolves it, creating brine that collects in nearby basins. The Orca Basin is one such place. At its eastern edge, a salt dome rises to the surface, and brine continuously seeps from it, flowing down into the depression where it has been accumulating for nearly 9,000 years. Scientists estimate that around 3.6 billion tons of salt have dissolved into the Orca Basin brine, and the process continues today at a rate of about 500,000 tons per year. But not all brine pools form through simple dissolution. Some are geothermally heated, created by processes associated with volcanic and hydrothermal activity. This is common in places like the Red Sea, where tectonic spreading is active. At the mid-ocean ridge running through the Red Sea, the Earth's crust is being pulled apart. Magma rises from below, creating new seafloor, and the heat from this magma warms the surrounding rocks. Seawater seeps down into the fractures created by the spreading, penetrating deep into the hot crust. There, it comes into contact with not only salt deposits, but also hot rock and minerals. The water dissolves salts and becomes enriched with metals and other chemicals. It is also heated to high temperatures. This superheated brine then rises back toward the seafloor, driven by convection. When it emerges, it is hot, dense, and chemically rich. It settles into depressions on the seafloor, forming hydrothermal brine pools. The Atlantis Code Deep is the most famous example. Discovered in the 1960s, it remains one of the hottest brine pools ever found, with temperatures in its lower layer reaching nearly 70 degrees Celsius. The heat is maintained by ongoing hydrothermal input from below, a slow but continuous flux of hot brine seeping up from the rocks beneath. There is also a third, less common way that brine pools can form, and that is through brine rejection, a process associated with sea ice. When seawater freezes, the ice crystals that form are made of pure water. The salt does not fit into the crystalline structure of ice, so it is expelled. This rejected salt sinks as dense, cold brine and can collect on the seafloor beneath the ice. This process is most common in polar regions, beneath Antarctic and Arctic sea ice, where cold, hypersaline pools can form in localized depressions. These polar brine pools are less well studied than their deep-sea counterparts, but they play an important role in ocean circulation, contributing to the formation of deep water masses that drive global currents. The deep-sea brine pools, however, are the ones that capture the imagination. They are hidden in darkness, accessible only by robotic submersibles, and they exist in some of the most remote and inhospitable places on the planet. Yet they are not rare. Scientists have identified brine pools in 3 major regions: the Gulf of Mexico, where at least dozens have been found, ranging from tiny puddles to the enormous Orca Basin; the Red Sea, where more than 25 individual brine pools or brine-filled deeps have been discovered, clustered along the central rift zone; and the Mediterranean Sea, where several large brine lakes sit at the bottom of the basin, more than 3,000 meters deep, formed from the dissolution of Messinian salt deposits. Each of these regions has its own character, its own history written into the chemistry and structure of its brine pools, and each pool, in turn, is unique. Even within the same region, no 2 brine pools are exactly alike. Their salinity, temperature, chemical composition, and even the life they support can vary dramatically. Some pools are relatively calm and stable, changing little over decades. Others are more dynamic, with active seeps and ongoing geological processes reshaping them. Some are layered with distinct convective zones stacked vertically, each with its own temperature and chemistry. Others are more uniform, a single body of dense liquid sitting quietly on the seafloor. To stand at the edge of one of these pools, to see the boundary between brine and seawater shimmering in the lights of a submersible, is to witness something deeply strange. It is water within water, yet separate. It is a boundary that should not exist, yet does. And as we will see, this boundary is not just a curiosity of physics and chemistry. It is a frontier between worlds, a place where death and life coexist in ways that challenge everything we thought we knew about the limits of survival. Let us travel now to the Gulf of Mexico, where some of the most studied and most dramatic brine pools on Earth can be found. The Gulf is a vast body of water, but beneath its surface lies a hidden landscape shaped by salt tectonics. The ancient Luan salt, deposited during the Jurassic, has been deformed over millions of years, creating a complex terrain of domes, ridges, and basins. In places, the salt has pushed upward so forcefully that it has created steep slopes and scarps on the seafloor. And in the depressions between these features, brine has pooled. The Orca Basin is the crown jewel of the Gulf's brine pools, both for its size and for the wealth of scientific knowledge it has provided. It sits on the continental slope, about 350 kilometers southwest of New Orleans, at a depth of around 2,400 meters. The basin itself is roughly 25 kilometers long and 6 kilometers wide, shaped like an elongated arc. At its deepest point, the brine layer is 220 meters thick, a column of hypersaline water resting on the sediments below. The brine in Orca Basin has a salinity of about 260 parts per thousand, roughly 7 times saltier than the ocean above it. Its density is around 1.3 grams per cubic centimeter, compared to 1.03 for normal seawater. This density difference of nearly 30% is enough to create a sharp, stable boundary that has persisted for thousands of years. The basin is anoxic, with no measurable oxygen below the halocline, and it contains high concentrations of methane and other dissolved gases. The temperature of the brine is slightly warmer than the surrounding deep water, around 5 degrees Celsius, due to the slow geothermal heat flux from below. Orca Basin was first identified in the 1970s, during surveys conducted by energy companies exploring for oil and gas. The unusual seismic characteristics of the basin caught the attention of geologists, and follow-up expeditions confirmed the presence of a massive brine pool. Since then, it has been visited repeatedly by research vessels and submersibles. In 1983, during leg 96 of the deep-sea drilling program, scientists drilled cores from the basin floor and made a remarkable discovery. They found gas hydrates, methane, locked into ice-like structures within the sediment, something that had not been documented before in the Gulf of Mexico at such depths. The sediments of Orca Basin hold another treasure, one that has made the basin invaluable to paleoceanographers. Because the brine is anoxic and isolated from the mixing currents of the ocean, anything that settles into it is preserved in exquisite detail. There are no burrowing animals, no worms or clams or crustaceans churning through the mud. The sediment layers lie undisturbed, stacked one atop another like the pages of a book, recording the history of the region over thousands of years. By coring into these sediments, scientists have been able to reconstruct the climate and oceanographic conditions of the Gulf of Mexico over the last 25,000 years. They have identified layers deposited by turbidity currents, underwater avalanches of sediment triggered by earthquakes or storms. They have found evidence of meltwater floods from the Laurentide ice sheet, the massive glacier that once covered much of North America, which sent pulses of fresh water down the Mississippi River and into the Gulf during the last deglaciation. The sediments even preserve the isotopic signatures of these events, allowing scientists to date them and understand their impact on the ocean and climate. But Orca Basin is not just a passive recorder of history. It is also a place of ongoing geological activity. The basin is surrounded by steep scarps and slopes, evidence of past landslides. In recent years, scientists studying high-resolution maps of the seafloor and seismic data have concluded that submarine landslides have repeatedly crashed into the Orca Basin, displacing the brine and creating massive waves within the pool. Imagine it. A landslide, perhaps triggered by the slow movement of salt beneath the seafloor, sends millions of tons of sediment and rock rushing down the slope. It hits the brine pool with tremendous force, and because the brine is so dense and viscous, it does not absorb the impact gently. Instead, it responds with waves, enormous internal waves that slosh back and forth within the basin. Scientists have calculated that these waves could reach heights of 100 to 360 meters, far larger than any tsunami ever recorded on the ocean surface. These waves do not break out into the open ocean. They are contained within the brine pool itself, reflecting off the basin walls, surging and subsiding over hours or days. But they are not harmless. If a wave were large enough, it could overspill the lowest edge of the basin, sending a pulse of hypersaline brine cascading into adjacent areas. This would be catastrophic for any marine life in the path of the brine, which is both toxic and oxygen-free. Researchers have found evidence that such overspill events have occurred in the past. Deposits of brine-derived sediment have been identified on the slopes above the basin, and the chemistry of these deposits matches the brine composition. The implications are profound. Brine pools are not static features. They are dynamic, shaped by ongoing geological processes, and they can interact with their surroundings in violent ways. Elsewhere in the Gulf of Mexico, other brine pools tell their own stories. In 2014, during an expedition led by the Ocean Exploration Trust, scientists using the remotely operated vehicle Hercules discovered a small brine pool at a depth of about 1,000 meters in an area called Green Canyon. This pool was immediately given a nickname by the researchers who found it. They called it the hot tub of despair, and later, the jacuzzi of despair. The name is darkly appropriate. The pool is roughly 100 feet in circumference and about 12 feet deep, a modest size compared to Orca Basin. But it is warm, with a temperature of around 19 degrees Celsius, significantly warmer than the surrounding water, which hovers just above freezing. The warmth comes from geothermal heat rising from below, giving the pool an almost inviting appearance. But inviting it is not. The salinity is around 200 parts per thousand, and the water is laced with methane and hydrogen sulfide. Any animal that enters the pool is in immediate danger. The footage captured by Hercules is haunting. The submersible approaches the edge of the pool, and the camera shows a distinct boundary, a shimmering interface between the brine and the seawater above. Surrounding the pool are dense beds of mussels, their shells packed tightly together, clinging to the rocks and sediment at the edge, and within the pool itself, the remains of animals that ventured too close. Crabs, eels, and other creatures lie motionless on the bottom, their bodies remarkably well preserved by the brine's high salt content. One crab, estimated to have been dead for 8 years, still had soft tissue intact, essentially pickled by the hypersaline water. The researchers watched as an eel swam too close and was immediately stunned upon entering the brine. It struggled, disoriented, and eventually died. Another predator, lurking nearby, seemed to use the pool as a hunting ground, darting in briefly to snag stunned prey before retreating to safety. The hot tub of despair is just one of many smaller brine pools scattered across the Gulf. Some are located at cold seep sites, places where methane and other hydrocarbons seep up from deep reservoirs beneath the seafloor. The methane provides energy for chemosynthetic bacteria, which in turn support dense communities of mussels, tube worms, and other organisms. The brine pools at these seeps are often ringed with life, oases in the otherwise barren deep sea. But the pools themselves remain deadly, traps for any creature unfortunate enough to fall in. The Gulf of Mexico brine pools are relatively cold compared to some others around the world. They are not geothermally heated in the same way as the Red Sea pools, but they are chemically rich, and they play an important role in the Gulf's ecosystem. They are also of great interest to the energy industry, because the salt structures that create them are often associated with oil and gas deposits. Understanding the geology of brine pools helps geologists locate and extract these resources. But for scientists interested in extreme environments and the limits of life, the Gulf's brine pools are laboratories. They offer a chance to study how organisms adapt to high salinity, low oxygen, and toxic chemicals, all in a natural setting that has existed for thousands of years. Now let us travel across the Atlantic, through the Suez Canal, and into the Red Sea, where an entirely different kind of brine pool awaits. The Red Sea is a young ocean, geologically speaking. It sits in a rift valley, where the African and Arabian tectonic plates are pulling apart at a rate of about 2.5 centimeters per year. This slow but relentless motion is tearing the Earth's crust open, creating new seafloor, and allowing magma to rise from the mantle below. The result is a landscape of volcanic ridges, deep basins, and hydrothermal activity. It is also a place where ancient salt deposits, formed during periods of isolation and evaporation, are being dissolved and mobilized by seawater, heated to extreme temperatures. The Red Sea contains more than 25 known brine pools, making it the region with the highest concentration of deep-sea brine pools on Earth. Most of these pools are located along the central rift zone in water depths of 2,000 to 3,000 meters. They vary widely in size, temperature, and chemistry, but nearly all of them are associated with hydrothermal activity. The most famous of these is the Atlantis to Deep, discovered in the early 1960s during a series of oceanographic expeditions that were among the first to explore the deep Red Sea. The Atlantis to Deep is not just a brine pool. It is a brine system, a complex of layered water masses stacked vertically within a depression on the seafloor. The lowest layer, called the lower convective layer, contains the hottest and saltiest water, with temperatures reaching 68 degrees Celsius and salinity around 257 parts per thousand. Above it are 3 additional layers, each slightly cooler and less saline, separated by thin interfaces where temperature and salinity change rapidly. The heat in Atlantis to Deep comes from below, from hydrothermal vents on the seafloor that continuously discharge hot, mineral-rich brine. Scientists have monitored the temperature of the deep for decades, and they have found that it has been slowly increasing, rising by about 0.1 degrees Celsius per year. This suggests that the hydrothermal system is still active, still delivering heat and chemicals into the deep. The chemistry of Atlantis Tekka Deep is extraordinary. It is not just hypersaline. It is also rich in dissolved metals. Iron, manganese, zinc, copper, and other elements are present in concentrations far higher than in normal seawater. As the hot brine cools and reacts with the seawater above it, some of these metals precipitate out, forming layers of metallic sediments on the seafloor. These sediments are so rich in metals that they have been considered as potential ore deposits, and there have been proposals over the years to mine them. The deep is also a biological hotspot. Despite its extreme conditions, the interface between the hot brine and the cooler seawater above is teeming with microbial life. Bacteria and archaea have adapted to thrive in this environment, using the dissolved gases and chemicals as energy sources. The temperature, which would kill most organisms instantly, is not a barrier to these extremophiles. They are among the most heat-tolerant organisms known on Earth, and studying them has provided insights into the limits of life and the kinds of adaptations that allow organisms to survive in seemingly impossible conditions. Nearby, but distinct in character, is the Discovery Deep, another large brine pool in the Red Sea. It was discovered around the same time as Atlantis Tekka Deep, and is located just a few kilometers away. Discovery Deep is cooler, with temperatures around 45 degrees Celsius, and its chemistry is different. It is less enriched in metals and has a higher concentration of hydrogen sulfide. The brine is still hot and hypersaline, but it appears to be fed by a different hydrothermal source, one that is less active or located deeper beneath the seafloor. A third major pool in the region is Kebrit Deep, which is even cooler, with temperatures around 23 degrees Celsius. Kebrit is unusual because it is not hydrothermally heated. Instead, it appears to be a cold brine pool, formed primarily by the dissolution of salt deposits without significant heat input. Its chemistry reflects this, with a lower metal content and a higher concentration of sulfide. Kebrit is also one of the most studied brine pools in the Red Sea, in part because its cooler temperatures make it somewhat more accessible for sampling, and in part because it hosts a rich microbial community that is easier to culture and study than the extreme thermophiles of Atlantis Tekka Deep. The Red Sea brine pools have been monitored for more than 50 years, making them some of the longest studied extreme environments on Earth. Expeditions in the 1960s, 1970s, 1980s, 1990s, and continuing into the 21st century have returned repeatedly to measure temperature, chemistry, and biology. This long-term dataset is invaluable. It has shown that the brine pools are remarkably stable over decadal timescales. The temperature of Atlantis Tekka Deep has increased slowly but steadily. The salinity has remained nearly constant. The layering of the water masses has persisted. This stability suggests that the hydrothermal systems feeding the pools are long-lived, and that the pools themselves have existed for thousands, perhaps tens of thousands of years. But stability does not mean unchanging. The pools are dynamic systems, with ongoing input of heat and chemicals from below and slow diffusive exchange with the overlying seawater. The sediments at the bottom of the pools are accumulating metals and organic matter, building up over time, and the microbial communities are constantly adapting, evolving and competing for resources in an environment that is both resource-rich and lethally extreme. In 2020, scientists aboard the research vessel Ocean Explorer made a new discovery that extended the range of brine pools in the Red Sea. They found a complex of brine pools in the Gulf of Aqaba, the narrow arm of the Red Sea that extends north between Saudi Arabia and Egypt. This was the first brine pool ever found in the Gulf of Aqaba, and it was unexpected. The Gulf of Aqaba is not part of the Central Rift Zone, and it was not thought to be a likely place for brine pools. But there they were, a main pool covering 10,000 square meters and 3 smaller pools nearby, each less than 10 square meters. The discovery was named the Neum Brine Pools, after the research initiative that funded the expedition. The Neum pools are located at a depth of 1,770 meters, close to the continental margin. They are cold brine pools, with temperatures similar to the surrounding seawater, and their chemistry suggests they were formed by the dissolution of evaporite deposits, rather than by hydrothermal activity. What makes the Neum pools especially valuable is their location. They are close to the coast, less than 2 kilometers from land. This means they are in a position to trap sediments washed off the land by rivers and flash floods. And because they are anoxic and hypersaline, those sediments are preserved in pristine condition, undisturbed by burrowing animals or bioturbation. When scientists cored into the floor of the main Neome pool, they recovered sediments representing at least 1,200 years of history. Within those sediments, they found layers deposited by turbidity currents, evidence of flash floods triggered by rare heavy rains in the desert. They found layers disturbed by seismic shaking, evidence of earthquakes that had occurred in the region. And they found layers of finer sediment that may have been deposited by tsunamis, carried inland by waves, and then washed back out to sea. This sedimentary record is incredibly valuable for understanding the natural hazards of the region. It provides a baseline for how often major earthquakes and floods occur, information that is crucial for planning infrastructure and development in the area. The Red Sea brine pools are windows into processes that are both ancient and ongoing. They show us what happens when seawater encounters buried salt and heat, when chemistry and geology conspire to create environments that are hostile to most life, but vital to the organisms that have adapted to them. They are also reminders that the ocean floor is not a static place. It is dynamic, shaped by forces we are only beginning to understand, and it holds secrets that have yet to be uncovered. And the Red Sea is not the only place in the world where brine pools have been found in deep basins far from hydrothermal vents. The Mediterranean Sea, that ancient body of water cradled between Europe, Africa, and the Middle East, hides its own collection of brine lakes formed from the same catastrophic event that nearly emptied the sea 6 million years ago. The Mediterranean brine pools are among the deepest on Earth. They sit at the bottom of the basin, more than 3,000 meters below the surface, in darkness and cold. Unlike the Red Sea pools, they are not heated. They are cold, with temperatures just a few degrees above freezing, but they are just as extreme in other ways. Their salinity can reach astonishing levels. One pool, the Discovery Basin in the eastern Mediterranean, was once thought to have the highest salinity of any natural water on Earth, with concentrations reaching more than 5 times that of seawater. Another pool, Lake Hephaestus, discovered more recently, may be even saltier. The Mediterranean pools are also anoxic, and their chemistry reflects the dissolution of Messinian evaporites, the salt layers left behind when the sea dried up during the salinity crisis. The dissolved salts are not just sodium chloride. They include magnesium, potassium, calcium, and sulfate, giving the brine a complex and variable chemistry. Different pools have different compositions, depending on which minerals are being dissolved and what diagenetic processes are occurring in the sediments below. The first Mediterranean brine pools were discovered in the early 1980s during surveys of the seafloor conducted as part of oceanographic research programs. One of the first to be identified was the Tyro Basin, located on the Mediterranean ridge. It was quickly followed by the discovery of the Bannock Basin and others. Since then, more than a dozen brine pools have been found in the eastern Mediterranean, each with its own characteristics. Some are relatively small, just a few hundred meters across. Others, like the Bannock Basin, are several kilometers long. All of them are located in topographic depressions, places where the seafloor dips down, allowing the dense brine to collect and remain isolated from the surrounding water. The Mediterranean pools are difficult to study. They are deep, remote, and challenging to sample. The high salinity of the brine can damage instruments, and the anoxic conditions mean that any biological sampling must be done carefully to avoid contamination. But the effort is worthwhile because these pools host unique microbial communities that have adapted to some of the most extreme conditions on Earth. The interface between the brine and the overlying seawater, the halocline, is especially rich in microbial life. Here, gradients of salinity, oxygen, and chemical composition create niches for different kinds of organisms. Some bacteria can tolerate only moderate salinity and live in the upper part of the halocline, where the brine is diluted by mixing. Others are extreme halophiles, organisms that require high salt concentrations to survive, and they live deeper, where the salinity approaches saturation. Some are methanogens, organisms that produce methane as a byproduct of their metabolism. Others are sulfate reducers, organisms that use sulfate as an electron acceptor in the absence of oxygen. Together, these microbes form a complex community that processes carbon, sulfur, nitrogen, and other elements, cycling them through the brine and the surrounding sediments. The Mediterranean brine pools are also important for another reason. They are analogues for ancient oceans. The chemistry and conditions in these pools may resemble those of Earth's early seas, before oxygen became abundant in the atmosphere. By studying the microbes that live in the brine pools and the biochemical pathways they use, scientists can gain insights into how life might have functioned in the distant past, and how it might function in similar environments on other planets or moons. This brings us to one of the most fascinating aspects of brine pools, and one that connects them to the search for life beyond Earth. Brine pools are not just curiosities, they are laboratories for astrobiology, places where we can study how life adapts to extreme conditions, and where we can test hypotheses about the kinds of environments that might support life elsewhere in the solar system. Jupiter's moon Europa and Saturn's moon Enceladus are both believed to have vast oceans beneath their icy crusts. These oceans are likely to be cold, dark, and potentially rich in salts and dissolved gases. If life exists in these subsurface oceans, it would face many of the same challenges that organisms face in Earth's brine pools: high salinity, low oxygen, darkness, and limited nutrients. By studying the microbes that thrive in brine pools, scientists can identify the kinds of metabolic strategies that might work in extraterrestrial oceans and the kinds of biosignatures, chemical or physical traces of life, that we might look for. Some researchers have gone so far as to suggest that brine pools on Earth are the closest analogues we have to the environments that might exist on ocean worlds like Europa. They are not perfect analogues, of course. The pressure, temperature, and chemistry are different, but they are similar enough to be useful, and they are accessible in ways that Europa's ocean is not, at least not yet. But let us return now to the brine pools themselves, and to the question that might seem most impossible: how can anything live in these places? They are toxic, they are anoxic, they are hypersaline, to the point of saturation. They contain hydrogen sulfide, methane, and heavy metals. They are, in every sense, death traps. And yet, life is there, not just surviving, but thriving. This is the paradox of brine pools, and it is one of the most remarkable stories in all of biology. To understand how life persists in brine pools, we must first understand what makes them deadly. When an animal enters a brine pool, several things happen almost immediately. The extreme salinity causes osmotic stress. Water is drawn out of the animal's cells, and salts flood in, disrupting the delicate balance of ions that cells need to function. The result is toxic shock. Proteins begin to denature, cell membranes lose their integrity, nerves and muscles cease to function properly. At the same time, the animal is breathing, or trying to breathe, but there is no oxygen in the brine. The gills or lungs extract nothing, and suffocation begins within seconds. And if the brine contains hydrogen sulfide or other toxic chemicals, those enter the body as well, poisoning enzymes and metabolic pathways. For most animals, the outcome is swift and inevitable: death. The brine does not break down the body in the way that normal seawater would. There are no scavengers in the brine, no worms or crabs or bacteria to consume the soft tissue. Instead, the high salt content preserves the body, pickling it in much the same way that salt preserves meat. Animals that have fallen into brine pools have been found decades later, their soft tissues still intact, their forms eerily recognizable. Crabs with their legs curled, eels with their mouths open, fish frozen in death, lying on the bottom of a pool that will hold them for centuries. But at the very edge of the brine pool, where the brine meets the seawater, something remarkable happens. Life finds a way. This boundary, the halocline, is not a sharp line. It is a gradient, a transition zone, where salinity changes from normal seawater at the top to full brine at the bottom. This gradient can be several meters thick, and within it conditions vary continuously. At the top, near the seawater, the salinity is low enough that many organisms can tolerate it. At the bottom, near the brine, only the most extreme specialists can survive. And in between, there is a zone where chemistry is just right, where methane and hydrogen sulfide from the brine meet oxygen from the seawater. This is where chemosynthesis happens. Chemosynthesis is the process by which certain bacteria and archaea can produce organic matter, food, without using sunlight. Instead of light, they use chemical energy, harvesting it from reactions like the oxidation of methane or hydrogen sulfide. These reactions release energy, and the microbes capture that energy and use it to convert carbon dioxide into sugars and other organic compounds. It is the same fundamental process as photosynthesis, but powered by chemistry instead of light. and it is the foundation of life at brine pools. At the edge of the pool, where the brine and seawater mix, methane bubbles up from below, carried by the brine or seeping through the sediments. Hydrogen sulfide does the same. And at the interface where these chemicals meet oxygen, chemosynthetic bacteria thrive. They form dense mats on the rocks and sediments, thick layers of microbial growth that can be meters wide. And these bacteria in turn support larger organisms. The most iconic of these are the mussels. In nearly every brine pool that has been studied, from the Gulf of Mexico to the Red Sea to the Mediterranean, scientists have found dense beds of mussels clinging to the edges of the pool. These are not ordinary mussels, they are specially adapted species, members of the genus Buthimodiolus, that have evolved to live in chemosynthetic environments. What makes them unique is that they do not feed by filtering plankton from the water the way most mussels do. Instead, they host chemosynthetic bacteria inside their bodies, in their gills. The bacteria live in the mussel's gill tissue, protected and nourished, and in return, they provide the mussel with food. The mussel takes in methane and hydrogen sulfide from the brine, along with oxygen from the seawater, and the bacteria use these chemicals to produce organic matter. The mussel then absorbs nutrients directly from the bacteria.
It is a symbiosis.
A partnership that allows both organisms to survive in an environment where neither could live alone. The mussel beds at brine pools can be enormous. At some sites in the Gulf of Mexico, beds of mussels stretch for tens or even hundreds of meters along the edge of the pool, their shells packed so tightly that they form a continuous crust. These mussels grow slowly, but they can live for many years, and over time the beds build up, layer upon layer of shells, creating structures that provide habitat for other organisms. Between the mussels you might find polychaetae worms, small crustaceans called amphipods, and snails. These animals are scavengers or predators, feeding on the mussels or on the organic matter that accumulates around them. They are not hosting bacteria themselves, but they benefit from the abundance of food that the chemosynthetic community provides. Also common at brine pools are tube worms, another iconic organism of chemosynthetic environments. These are not the same as the giant tube worms found at hydrothermal vents, though they are related. The tube worms at brine pools belong to genera like Lamellobrachia and Sepiophila, and they can grow to be more than a meter long. Like the mussels, they host chemosynthetic bacteria, but the bacteria live in a specialized organ called the trophosome, located inside the worm's body. The worm has no mouth and no digestive system. It relies entirely on the bacteria to produce food. The worm's bright red plume, which extends from the top of its tube, is filled with blood vessels that carry hydrogen sulfide and oxygen to the bacteria. The worm essentially farms the bacteria, providing them with the chemicals they need and harvesting the nutrients they produce. Tube worms can form dense thickets at brine pool edges, their white tubes standing upright from the seafloor like a strange forest. These structures provide habitat for many smaller organisms, including shrimp, copepods, and anemones, all of which live among the tubes and feed on the organic matter produced by the worms and their bacteria. The chemosynthetic community at a brine pool is in many ways an oasis. The deep sea is generally a desert, a vast, food-poor environment where most organisms survive on the scraps that drift down from the surface, the so-called marine snow. But at a brine pool, there is a steady supply of chemical energy, and the organisms that can tap into that energy grow large and reproduce quickly. This abundance attracts predators. Crabs, hagfish, and eels are common visitors to brine pool edges, where they hunt the mussels and worms. Larger fish and even sharks have been observed circling above the pools, taking advantage of the concentration of prey. Some predators have learned to use the brine itself as a tool. Eels, for instance, have been observed stunning small prey by driving them into the brine, where the toxic water immobilizes them, making them easy to catch. But the predators must be careful. If they venture too far into the brine, they too will be overcome. The boundary between life and death at a brine pool is razor thin, a matter of centimeters. Step across it, and you die. Stay on the edge, and you thrive. It is a stark reminder of how finely tuned life can be to its environment, and how even small changes in chemistry or geography can mean the difference between survival and extinction. The microbial life within the brine itself is even more remarkable. For decades, scientists believed that the brine pools were completely sterile, that nothing could live in such extreme conditions. But as sampling techniques improved, and as scientists began to culture and sequence the DNA of microbes from brine samples, a different picture emerged. The brine is not sterile. It is teeming with life, but that life is invisible to the naked eye, composed entirely of microorganisms, bacteria and archaea that have adapted to conditions that would be instantly lethal to any other form of life. These are extremophiles, organisms that not only tolerate extreme conditions but require them, and within the brine pools, they have been given a name that reflects the multiple extremes they face. They are polyextremophiles, organisms that survive high salinity, high temperature or extreme cold, anoxia, high pressure, and high concentrations of toxic chemicals, all at the same time. The microbial communities in brine pools are incredibly diverse. Different pools, and even different layers within the same pool, host different communities, shaped by the specific chemistry and conditions of each environment. In the hot metal-rich brine of Atlantis Tseken deep in the Red Sea, scientists have found bacteria and archaea that can tolerate temperatures up to 70 degrees Celsius and salinity more than 7 times that of seawater. Some of these microbes are extreme halophiles, members of the domain archaea that require high salt to stabilize their proteins and membranes. Others are thermophiles, organisms that thrive at high temperatures and whose enzymes are adapted to function in conditions that would denature most proteins. In the cooler, sulfide-rich brine of Kebrit Deep, the microbial community is dominated by sulfate-reducing bacteria, organisms that use sulfate as an electron acceptor in the absence of oxygen. These bacteria play a key role in the sulfur cycle, converting sulfate into hydrogen sulfide, which then provides energy for other organisms at the interface. In the Mediterranean brine pools, where the salinity can reach even higher levels, scientists have found archaea that are among the most salt-tolerant organisms known. Some of these can survive in brines that are nearly saturated, where the salt concentration approaches the point of crystallization. The adaptations that allow these microbes to survive are remarkable. At high salinity, water is scarce. Even though the brine is liquid, the high concentration of dissolved salts makes it difficult for cells to retain water. To cope, halophilic microbes accumulate high concentrations of compatible solutes, organic molecules that stabilize proteins and prevent water loss. Some also accumulate potassium ions inside their cells to balance the sodium outside. At high temperatures, proteins and cell membranes become unstable. Thermophilic microbes produce heat shock proteins and chaperones that help refold damaged proteins, and they synthesize membrane lipids that remain stable at high temperatures. In anoxic conditions, organisms must find alternative ways to generate energy, since they cannot use oxygen for respiration. Many brine pool microbes use anaerobic respiration, using compounds like sulfate, nitrate, or even metals like iron or manganese as electron acceptors. Others use fermentation, breaking down organic matter into simpler compounds and extracting a small amount of energy in the process. The microbes in brine pools are not just surviving, they are active, carrying out metabolic processes that shape the chemistry of the brine and the sediments. They are cycling carbon, nitrogen, sulfur, and metals, transforming these elements in ways that affect the entire system, and they are producing compounds that are of interest to biotechnology. Enzymes from brine pool microbes, called extremozymes, are stable and functional under conditions that would destroy most enzymes. They can tolerate high salt, high temperature, and extreme pH, making them useful for industrial processes like chemical synthesis, biofuel production, and wastewater treatment. Some of these enzymes have already been isolated and characterized. A nitrolase from Atlantis to Tseken Deep, for instance, is unusually stable and can tolerate heavy metals, properties that make it valuable for biotechnology applications. A mercuric reductase from the same pool can detoxify mercury, a property that could be useful for bioremediation. Researchers are actively screening brine pool microbes for new enzymes and for genes that encode novel biochemical pathways, and the potential applications are vast. But the microbes are also scientifically valuable for what they tell us about the limits of life. By studying how they adapt to extreme conditions, we learn what is possible, what kinds of environments can support life, what kinds of metabolic strategies can work when resources are scarce and conditions are harsh. This knowledge is not just academic, it informs our search for life on other planets and moons, and it helps us understand how life on Earth might have begun, in an early ocean that was likely hotter, more acidic, and more chemically extreme than the ocean of today. There is also a darker side to the story of brine pools, a reminder that the deep sea is not immune to human impact. As submersibles and remotely operated vehicles have explored brine pools, they have occasionally found evidence of pollution, debris that has made its way to the seafloor and come to rest at the edges of the pools. Plastic, metal, and other waste have been observed at some sites, a sobering indication that even the most remote and extreme environments are not beyond the reach of human activity. The chemistry of brine pools can also be affected by changes in the ocean. If the temperature of the deep sea rises, if ocean circulation patterns shift, if the input of fresh water from rivers or melting ice changes the salinity of the ocean, these changes could alter the stability and chemistry of brine pools. We do not yet know how resilient these systems are to change, or what the consequences might be for the organisms that depend on them. What we do know is that brine pools are fragile in some ways, even as they are remarkably stable in others. They are shaped by processes that operate over millennia, but they can be disturbed by events that happen in an instant. A submarine landslide, a sudden influx of sediment, a change in the hydrothermal flux from below. All of these can reshape a brine pool, and all of them have happened in the past. As we continue to explore the ocean and to study brine pools, we are also learning to appreciate them not just as scientific curiosities, but as integral parts of the deep sea ecosystem. They are not isolated. They are connected to the surrounding ocean by currents, by the animals that visit them, and by the chemicals that diffuse out of them. They are part of a larger system, and they play a role in the biogeochemical cycles that regulate the chemistry of the ocean and the atmosphere. The discovery of brine pools has been a slow and painstaking process, driven by advances in technology and by the persistence of scientists and explorers who ventured into the deep despite the challenges. The first brine pools were found by accident, during surveys of the seafloor conducted for other purposes. As instruments became more sophisticated, and as submersibles became capable of reaching greater depths and staying down longer, the number of known brine pools grew. The development of remotely operated vehicles, or ROVs, was a turning point. These robotic submersibles, tethered to a ship by a cable that carries power and communication signals, can descend to the deepest parts of the ocean and send back high-definition video and data in real time. They are equipped with lights, cameras, sensors, and manipulator arms that allow them to collect samples and conduct experiments on the seafloor. Without ROVs, much of what we know about brine pools would still be unknown. The Ocean Exploration Trust, founded by oceanographer Robert Ballard, has been at the forefront of brine pool exploration. The Nautilus, the organization's research vessel, and its ROV Hercules have visited brine pools in the Gulf of Mexico and elsewhere, capturing footage that has been streamed live on the internet and watched by millions of people around the world. These expeditions have revealed not just the brine pools themselves, but the life around them and the geological processes that shape them. The footage is often breathtaking. The ROV descends through darkness, its lights cutting through the blackness, revealing glimpses of the seafloor. And then, suddenly, there it is, a pool, a boundary, a shimmering surface. The camera zooms in, and you can see the ripples, the disturbances caused by currents. You can see the mussels, clinging to the edge. You can see the eels, swimming cautiously, and the crabs, scuttling across the rocks. And you can see the bodies of animals that ventured too close, lying still in the brine, preserved by the salt. The scientific value of these expeditions cannot be overstated. Every dive reveals something new, a species that has not been seen before, a chemical process that was not expected, a feature of the brine pool that changes our understanding of how these systems work, and the public engagement is equally important. By sharing the exploration in real time, the Ocean Exploration Trust and similar organizations have brought the deep sea into homes and classrooms, inspiring a new generation of scientists and ocean advocates. Other research vessels have also made important contributions. The Ocean Explorer, a state-of-the-art research and media vessel, discovered the Innuan brine pools in the Gulf of Aqaba in 2020. The expedition was part of a broader effort to survey and understand the Red Sea, and the discovery of the brine pools was a highlight. The ship was equipped with submersibles, ROVs, and a suite of sensors that allowed the team to map the seafloor, sample the water and sediments, and document the life around the pools. The data from that expedition, including sediment cores that recorded 1,200 years of environmental history, have been published in scientific journals and are being used to inform hazard assessments and development planning in the region. Academic research institutions around the world have also contributed to brine pool science. Universities in the United States, Europe, the Middle East, and elsewhere have sent expeditions to study brine pools, often in collaboration with government agencies and international organizations. These studies have included everything from microbiology to geochemistry to sedimentology, and they have produced hundreds of scientific papers that collectively form the foundation of our understanding of these environments. But there is still so much we do not know. Many brine pools have been identified but not thoroughly studied. Others almost certainly remain undiscovered, hidden in remote parts of the ocean that have never been surveyed. And even the brine pools that have been studied extensively still hold mysteries. How old are they? How stable are they over long time scales? How do the microbial communities evolve and adapt? What triggers the landslides and other disturbances that reshape them? These are questions that will require years, perhaps decades, of continued research to answer. As we bring our journey to a close, it is worth reflecting on what brine pools teach us. They are, in many ways, a reminder of the ocean's vastness and complexity. The ocean is not a uniform body of water. It is a mosaic of environments, each with its own chemistry, its own physics, its own biology. Brine pools are just one piece of that mosaic, but they are a piece that challenges our assumptions and expands our understanding of what is possible. They teach us that life is resilient and adaptable, capable of finding footholds in places that seem utterly inhospitable. They teach us that the boundaries between environments are not always sharp, that gradients and interfaces can be as important as the environments themselves. They teach us that geology and biology are intertwined, that the rocks and sediments beneath our feet are not just a passive stage but an active participant in the drama of life. And they teach us humility. For all our technology and knowledge, the ocean still holds secrets. There are still places we have not been, processes we do not understand, organisms we have not discovered. The brine pools are a reminder that the deep sea is not just a frontier of exploration, but a frontier of science, a place where every question answered raises new questions, and where the act of looking deeper reveals not just what is there, but how little we truly know. And so, as you rest tonight, let yourself drift down through the layers of the ocean, through the sunlit surface, through the twilight zone where light fades to darkness, through the midnight zone where no light penetrates, down, down, down to the abyssal plain where the pressure is immense and the cold is absolute. And there, in a depression on the seafloor, imagine a pool, still and quiet, its surface unbroken except for the slow ripples of a deep current. Around its edge, a garden of mussels and tube worms, fed by invisible bacteria, sustained by chemicals rising from the earth. And within the pool, dissolved in the brine, ancient salts from seas long vanished, carried through time and held in suspension, waiting. This is the world of brine pools, hidden, extreme, beautiful in its strangeness. A reminder that even in the darkest, most inhospitable corners of the planet, there is life, there is chemistry, there is story. And perhaps as you close your eyes and let sleep come, you will dream of these still lakes in the deep, of the boundaries between worlds, of the life that clings to edges, of the mysteries that remain, waiting to be discovered. Sleep well, the ocean is vast, and there is always more to explore. But for now, rest, let the currents carry you, and know that somewhere far below the brine pools are still there, unchanged, unhurried, holding their secrets in the dark.